Hello class, let's continue to learn chapter 3. Last time we have learned about the actinomycetes. In this lecture, we are going to learn about the Mycoplasma, Rickettsia, and Chlamydia. Members of molecules are commonly called mycoplasmas. These bacteria are characterized by the absence of cell walls, their small genomes, and simplified metabolic pathways. Mycoplasmas are polymorphic, varying in shape from spherical or pear-shaped microbes from 0.3 to 0.8 microns in diameter to slender branched or helical filaments. Some mycoplasmas have a specialized terminal structure that projects from cell and gives them a flask or pear shape. This structure aids in attachment to eukaryotic cells. Mycoplasmas are among the smallest bacteria capable of self-production. As we mentioned before, sterols are an essential component of the mycoplasma membrane where they may facilitate osmotic stability in the absence of a cell wall. When grown on agar, most form colonies with a fried egg appearance because they grow into the agar surface and the center while spreading outward on the surface and the cloning edges. Mycoplasmas are remarkably widespread and can be isolated from animals, plants, soil, and even compost piles. Although their complex growth requirements can make their growth in pure cultures difficult, at least 10% of the mammalian cell culture in use are probably contaminated with mycoplasmas. This seriously interferes with tissue culture experiments and has resulted in the development of commercially available rapid tests for detection of mycoplasmas in cell culture. In animals, mycoplasmas colonize mucous membranes and joints, and often are associated with diseases of the respiratory and urogenital tracts. Spiroplasmas have been isolated from insects, ticks, and a variety of plants. They cause disease in citrus plants, cabbage, broccoli, corn, honeybees, and other hosts. In humans, Ureaplasma urealecticum and Mycoplasma hominis are common parasitic microorganisms of the genital tract. The genus Rickettsia contains rod-shaped, coccoid or polymorphic cells with typical gram-negative walls and no flagella. Although their size varies, they tend to be very small. Members of the genus Rickettsia are 0.3 to 0.5 microns in diameter and 0.8 to 2 microns long. Most are pathogens that alternate between a mammalian host and a blood-sucking arthropod. All species are parasitic or mutualistic. The parasitic forms grow in vertebrate astrocytes, macrophages, and vascular endothelial cells. Some also live in blood-sucking arthropods, such as fleas, ticks, mites, or lice, which serve as vectors or primary hosts. Rickettsia have lost the genes encoding glycolytic pathway enzymes, so they can't use glucose as an energy source. Instead, they oxidize glutamate and TCA cycle intermediates. Several important human diseases are caused by Rickettsia. Among these are Rocky Mountain spotted fever caused by Rickettsia rickettsii and endemic typhus caused by Rickettsia typhi. These microbes enter host cell by inducing phagocytosis, but immediately escape the phagocytic vacuole to reproduce by binary fission in the cytoplasm. They continue to reproduce until the host cell bursts, releasing bacteria to infect other host cells. As figure shown, Rocky Mountain spotted fever is characterized by the sudden onset of a headache, high fever, chills, and a skin rash that initially appears on the ankles and wrists, and then spreads to the trunk of the body. The symptoms reflect the accumulation of lyased host cells and toxic effect of Rickettsia cell walls. If the disease is not treated, Rickettsia can destroy the blood vessels in the heart, lungs, or kidneys and cause death. Chlamydia are gram-negative obligate intracellular parasites. This means they must grow and reproduce within host cells. Members of the genus Chlamydia and Chlamydophila are non-motile, coccoid bacteria ranging in size from 0.2 to 1.5 microns. Chlamydia metabolism is very different from that of other bacteria. It had been thought that chlamydia cannot catabolize carbohydrates or synthesize ATP. The chlamydia can be cultivated in laboratory animals, in cell cultures, or in the yolk sac of embryonated chicken eggs. Chlamydia reproduction is unique. As figure shown, reproduction begins with attachment of an alimentary body to the host cell surface. Alimentary bodies are 0.2 to 0.6 microns in diameter, contain electron-dense nuclear material and a rigid cell wall. Host cells become infected with alimentary bodies by endocytosing them. An alimentary body is held in an endotherm that evades fusing with lysosomes. The alimentary bodies differentiate into a reticulate body and undergoes binary fission, which continues until the host cell dies. Reticulate bodies are 0.5 to 1.5 microns in diameter and have less dense nuclear material and more ribosomes than alimentary bodies. Their walls are also more flexible. After 20 to 25 hours, reticulate bodies begin to differentiate into infectious alimentary bodies. The host cell lyses and releases alimentary bodies 48 to 70 hours after infection. Chlamydia trachomatis is the best-known pathogen of the group and responsible for more than one major disease. Chlamydia trachomatis is responsible for the most common sexually transmitted disease. It is also the most common cause of preventable blindness in the world. As figure shown, repeated infection with Chlamydia trachoma cause chronic inflammation. The eyelid has been pulled back to show the inflammatory nodules that are in contact with cornea. The abrasion caused by this damages the cornea and makes it susceptible to secondary infections. Okay, that's all for today's lesson.